have you ever thought about what happens when a medical treatment meant to cure a disease accidentally forces that disease to become smarter and, faster and deadlier?
It's a chilling paradox, honestly.
Right. You think you've cornered it, but the intense pressure you apply actually teaches the disease how to survive in a much more terrifying form.
Exactly. It's one of the most critical challenges we face in modern oncology. We are essentially watching forced evolution happen fast forward right inside the human body.
It's wild. So today we are looking at some really fascinating 2026 research from NPJ Precision Oncology about how prostate cancer pulls off this exact trick.
Yeah, it's a survival tactic called lineage plasticity.
Right. Basically, the cancer literally shapeshifts into a highly aggressive treatment-resistant form called neuroendocrine prostate cancer, or NEPC.
And the researchers actually managed to catch the cancer in this hidden transition phase, which they call early neuroendocrine differentiation, or early NED.
So that's the mission for today's deep dive. We're going to explore the spatial technology they use to finally see this transition, dissect this 6-gene alarm system that flags it, and look at the biological engine, this specific cellular switch that drives the whole process.
We are getting into the weeds on this one for sure.
Okay, let's unpack this.
Yeah.
How does a cancer cell literally change its identity to avoid being killed?
Well, to understand that, we need to look at the clinical reality. The study brings up real clinical scenarios like patient 040.
Right, because the stakes here are incredibly high.
Exactly. So this is someone who had standard prostate cancer and received androgen deprivation therapy, but 14 months later, the cancer recurred locally.
Which raises the question, what exactly happened in that 14-month window?
To figure that out, we have to look at the mechanics of that initial treatment. Androgen deprivation therapy, or ADT, is a foundational treatment.
Because prostate cancer needs testosterone, right?
Right. Prostate adenocarcinoma cells, the most common type, are fundamentally addicted to androgens, primarily testosterone. The testosterone binds to androgen receptors in the cell, which then act as transcription factors.
So they basically signal the cell to grow and divide.
Precisely. So ADT is basically starving the cancer by cutting off that specific chemical fuel supply.
And initially, it works, right?
Oh, yeah. Initially, a massive number of cancer cells undergo apoptosis, programmed cell death, because they are starved.
Right,
But a tumor is made of millions of cells, and they are fighting for their lives. Under this intense selective therapeutic pressure, a subset of those adenocarcinoma cells initiates an epigenetic reprogramming.
Wow.
Yeah, they undergo a transdifferentiation process.
Meaning they aren't just mutating slightly, they are actively changing their fundamental cellular identity just to survive the starvation.
Yes. They enter that early NED phase we mentioned. They are basically rewriting their internal software so they no longer rely on the androgen receptor pathway at all.
And then they complete the transformation.
Right. Eventually they complete it and become treatment-related neuroendocrine prostate cancer, or TNEPC. And the TNEPC phenotype is brutal.
How so?
Well, unlike localized gland cells, neuroendocrine cells are inherently secretory and migratory. This leads to widespread visceral metastases, meaning it rapidly spreads to major organs like the liver and lungs.
And it develops rapid resistance to other therapies along the way, too. But there's a diagnostic problem here as well, isn't there?
A massive one.
The study mentions they stopped producing PSA, or prostate-specific antigen. But PSA is like the primary biomarker doctors use to track prostate cancer progression.
Exactly, because PSA production is driven by the androgen receptor.
Oh, I see.
So if the cancer cell has completely abandoned the androgen receptor to survive, it just stops making PSA. So the patient's blood work might look perfectly fine.
That is terrifying.
The standard diagnostic alarms stay completely silent while this highly aggressive variant rapidly spreads.
To map this out visually for you guys, it's essentially like a fugitive on the run.
Yeah.
The police, which would be the androgen deprivation therapy, set up roadblocks all over the city, specifically looking for a guy in a red suit representing the adenocarcinoma.
Right.
The fugitive hits a roadblock, realizes he's trapped, so he ducks into an alley, strips off the red suit, and puts on a blue suit, which is the neuroendocrine cell.
And because he's in the blue suit, he just walks right through the roadblock. The therapy targets a specific vulnerability, so the cancer simply sheds the vulnerability entirely.
But wait, if he's just changing his disguise in the alley, why has it been so incredibly hard for oncologists to catch these cells during the costume change?
That's the tricky part.
Like, why can't we just look at a biopsy and see the transition happening?
Because morphologically, under a standard microscope, a cell in the early stages of this transition still looks exactly like an adenocarcinoma cell.
Oh, really?
Yeah, the changes are happening deeply at the molecular and genetic levels long before they physically change the shape of the cell. Finding this early NED phase has been a massive diagnostic blind spot because our traditional visual tools just couldn't perceive the internal software update happening inside the cell.
Okay, before we get into the lab tech they used to finally see this software update, I want to do a quick recap to make sure we're fully wrapping our heads around this timeline.
Good idea.
So if I'm tracking with you, there are 3 main steps here for you to remember. First, prostate cancer relies on androgens, so we use ADT to cut off the fuel.
Right.
Second, instead of dying, the cancer survives the starvation by shape-shifting its internal identity into neuroendocrine cells.
Exactly.
And third, this transition creates TNEPC, a deadly variant that is practically invisible to standard PSA tests.
That is the exact clinical progression.
Have you ever considered that the very drugs designed to corner a disease might actually be the ultimate test that forces it to evolve into a super predator?
It really completely reframes how we view targeted therapy. We aren't just killing cells, we are inadvertently steering their evolution.
Which brings us to the technology. If these cells look completely normal under a microscope during the transition, how did the researchers in this 2026 study actually see into that dark alley?
They used a combination of tools. The paper mentions single-cell RNA sequencing and GeoM's digital spatial profiling, or DSP.
Okay, let's start with single-cell RNA sequencing. How does that help?
So single-cell RNA sequencing, or cRNA-seq, allows us to look at the transcriptome, the active RNA, being produced in individual cells rather than just a bulk sample.
Which gives you a lot of detail.
Right, it reveals the incredible heterogeneity of a tumor. We can see that cell A is behaving very differently from cell B, but cRNA-seq has a major flaw.
But what is it?
To do it, you have to take the tumor tissue and dissociate it. You chemically break it apart so the cells are floating individually in a liquid.
So you know exactly what each cell is doing, but you lose all the geographical context.
Exactly.
You have no idea which cell was sitting next to which cell in the actual tumor. It's like throwing a whole city into a blender.
That's a great way to put it.
You can taste the smoothie and know exactly what ingredients are in it, but you don't know who lived in which neighborhood.
And that's exactly where GeoM's digital spatial profiling comes in. DSP preserves the map.
How so?
The researchers take incredibly thin slices of intact tumor tissue and place them on a slide. Then they wash the tissue with oligonucleotide tags. These are essentially tiny molecular barcodes that bind to specific RNA targets inside the tissue.
But if they're just washing the whole slide in barcodes, how do they differentiate the cells?
They use specific fluorescent protein markers alongside the barcodes. In this study, they used a marker called CGA, or chromogranin A, which physically lights up the neuroendocrine cells, and NKX3.1, which lights up the adenocarcinoma cells.
Yeah.
So now you have a physical map glowing with different colors.
That sounds incredible.
It is. When the researchers identify a specific microscopic cluster of cells they want to analyze, say a cluster where red and blue cells are mixing, they shine an ultra-precise UV laser onto just that exact spatial region.
And what does the laser do? Does it cut the tissue out?
No, it's much more elegant. The UV light physically breaks a photocleavable bond connecting the barcode to the tissue. It basically snips off the oligonucleotide tags just in that microscopic area illuminated by the laser.
Oh, wow.
Those freed tags are then sucked up into a microcapillary tube and sequenced.
That is mind-blowing. So they aren't destroying the map. It's like having a highly advanced satellite view of the city. You see the houses, and the laser lets you tap the phone lines of just one specific house to hear exactly how those specific cells are communicating.
Yes, and when they tapped those localized phone lines, they found something revolutionary.
What was it?
They discovered that neighboring cells within the exact same tumor microenvironment, or TME, shared remarkably similar gene expressions regardless of their actual cell type.
Wait, regardless of their type?
Right. If a standard adenocarcinoma cell was sitting physically right next to a neuroendocrine cell, its internal genetic machinery started to mirror the neuroendocrine cell's machinery.
Here's where it gets really interesting. If the neighbors are whispering, are you saying the normal cancer cells are essentially being peer pressured into changing their identity just because of who they are sitting next to?
That is the most profound implication of the spatial data. This peer pressure happens through paracrine signaling.
Which is?
The neuroendocrine cells secrete chemicals, things like interleukins or neuropeptides, into the extracellular matrix. These chemicals drift over to the neighboring adenocarcinoma cells and bind to their receptors.
So they're directly influencing them.
Right. It acts as a localized environmental pressure, operating on a mechanism similar to quorum sensing in bacteria, where cells coordinate their behavior based on the density of their population.
So the neuroendocrine cells are literally whispering to the adjacent cells, telling them to start the transition.
They are actively recruiting them.
Okay, let's pause and summarize what we've discovered about this molecular neighborhood. First, spatial profiling preserves the neighborhood context, letting researchers read the genetic activity without destroying the map. And second, by keeping that map intact, they revealed that early NED relies on local intercellular communication. The cancer cells are peer pressuring each other.
Exactly.
So, listener, think about this. If a tumor is functioning as a corrupted communicating community like this, how could we therapeutically shut down their communication network before that bad idea spreads to the rest of the tissue?
It heavily suggests that disrupting the tumor microenvironment's communication network is just as vital as directly attacking the cancer cells themselves.
So we know how they are talking. Now let's dig into the alarm system the researchers built to catch them in the act.
Right, the gene signature.
The paper outlines a massive intersectional analysis of multiple data sets to find the specific genes that spike during this early transition. They narrowed it down to 6 upregulated marker genes.
Yes, FMN2, APLP1, SCG2, SCG3, CHGB, and RMBP2. We need to look at the mechanical function of these 6 genes because they prove that the cell isn't just surviving, it is fundamentally rebuilding its internal architecture.
Let's break them down. What do CHGB, SCG2, and SCG3 actually do?
These 3 are established secretory granule markers. Neuroendocrine cells, by their nature, secrete hormones and peptides to communicate and drive growth.
Okay.
To do that, the cell needs physical shipping containers, secretory granules, to package and transport these chemicals out of the cell. The upregulation of these 3 genes means the cancer cell is heavily investing in building up a massive secretory capacity.
Okay, so that's the shipping department. What about FMN2?
FMN2 regulates actin cytoskeletal stability.
The scaffolding.
Right, the physical protein scaffolding inside the cell. For an adenocarcinoma cell to become a migratory neuroendocrine cell, it has to physically restructure its shape. It has to tear down its old scaffolding and build new structural supports to accommodate its new function and mobility.
It's literally rebuilding its own skeleton.
Yeah.
And the last 2, APLP1 and RMBP2.
This is where the biology gets truly unsettling. RIMBP2 and APLP1 are genes critical for synaptic vesicle release and neuronal differentiation.
Wait, neuronal? Like in the brain?
Yes. These are genes typically used by brain neurons to wire themselves together and transmit electrical and chemical signals across synapses.
Why on earth would a prostate cancer cell need brain neuron genes?
To acquire neuron-like structural plasticity, the tumor is wiring itself into the body's nervous system or creating neural-like networks within the tumor microenvironment to accelerate that paracrine signaling we discussed earlier.
That is terrifying.
It is a highly advanced adaptation.
To help visualize this, it's essentially like a factory being retooled in the dark. FMN2 is the new physical scaffolding holding the building up. The SCGs and CHGB are the new shipping boxes being manufactured to send out localized chemical packages.
Right.
And RMBP2 and APLP1 are the new electrical wires being run through the walls to connect it all together.
That retooling has immense clinical consequences. The researchers validated the 6-gene signature using bulk RNA sequencing on a cohort of 86 patients, and then cross-referenced it with a massive external cohort of 498 patients from the Cancer Genome Atlas.
So what does this all mean for the patient? Is this 6-gene score just a neat biological fun fact, or is this a literal alarm system that could change how we prescribe drugs?
It's an alarm system that directly predicts survival. They proved that a risk score based on the expression levels of these 6 genes correlates with a significantly shorter progression-free survival.
Wow.
Patients whose tumors express high levels of this 6-gene alarm system relapse much faster. But it also provides a potential roadmap for personalized chemotherapy.
How does it do that?
The data suggests that high expression of APLP1 is linked to resistance to docetaxel, which is a very common chemotherapy.
So if a patient has high APLP1, giving them docetaxel might be a complete waste of time or even harmful.
Exactly. Conversely, they found that high expression of FMN2 and SCG2 is linked to sensitivity to bortezomib, a completely different class of drug known as a proteasome inhibitor.
So to synthesize this segment, the researchers identified a specific 6-gene signature, which acts as a dual-purpose tool. It's a prognostic risk score that flags the early transition and a potential guide for personalized chemotherapy.
Spot on.
So if you had a genetic alarm system that could warn you a tumor was retooling its factory, how would you change the treatment plan? Would you still hit it with the standard frontline drugs or completely pivot your strategy?
This molecular insight demands that we pivot. But to truly stop the transition, we have to understand what turns the power on in that factory.
Right.
We know the genes that get activated, but what flips the switch?
Well, let's take a beat for our second 5-minute recap before we get into the engine of this thing.
Great. So quick bullet points for you. Number 1, GeoMx DSP allowed researchers to map the tumor microenvironment spatially without destroying the geography. Number 2, cancer cells peer pressure each other through intercellular communication. And number 3, a specific 6-gene signature FMN2, APLP1, SCG2, SCG3, CHGB, and RMBP2 accurately flags this early transition and predicts poor survival.
Perfect. So returning to that switch, the researchers found the biological engine driving this entire costume change, and it comes down to a signaling pathway called the Hedgehog pathway.
Right. The Hedgehog pathway is deeply conserved in biology. It plays a massive role in embryonic development. It tells cells where to go and what to become when an embryo is forming. But in healthy adult tissues, it is usually heavily suppressed or completely turned off.
But cancers are famous for hijacking embryonic pathways to fuel their own chaotic growth, right?
Exactly. Let's trace the physical mechanics of this pathway based on the study. It begins with a signaling protein, a ligand called SHH, which stands for sonic hedgehog.
I always forget scientists named a crucial embryonic protein after a video game character.
It's a classic quirk of genetic nomenclature. But in this early NED phase, the cancer cells begin producing highly elevated levels of this SHH ligand.
Okay.
This SHH protein gets secreted and travels to the cell membrane, where it binds to a specific transmembrane receptor called PTCH1.
Okay, so SHH binds to the PTCH1 receptor on the cell surface. What happens inside the cell?
Normally, when the pathway is off, PTCH1 acts as a brake. It actively inhibits another transmembrane protein called SMO, or smoothened. But when the SHH ligand binds to PTCH1, it changes its shape, lifting that inhibition.
So SMO is suddenly released.
Yes, the brake is taken off.
Where does SMO go?
SMO initiates a complex intracellular cascade that ultimately stabilizes and upregulates a transcription factor called GLI1. GLI1 then travels into the nucleus of the cell, binds to the DNA, and actively turns on the genes necessary for the cell to undergo neuroendocrine transdifferentiation.
Got it.
The study found highly elevated levels of the SHH ligand, the PTCH1 receptor, and the GLI1 protein specifically localized in the tissues undergoing this early transition. But here's the crucial twist in the paper, and this is the part that really stood out to me. The Hedgehog pathway is only active during the early NED phase.
Right.
By the time the cancer reaches the late stage, full-blown TNEPC, like patient 043 or 040, progressing to their recurrence, the Hedgehog pathway goes completely silent.
That is the pivotal discovery of this research. It highlights a phenomenon we call stage-specific regulation. In late-stage TNEPC, a completely different biological engine takes over. It's called the Myc pathway.
And what does the Myc pathway do differently?
Myc pathway is a notorious oncogene. It doesn't care about changing the cell's identity. It drives sheer, unadulterated cellular proliferation.
Just rapid growth.
Exactly. It upregulates cell cycle regulators like CDK1, forcing the cell to divide continuously. The cancer stops focusing on the complex energy expenditure of transdifferentiation and focuses entirely on multiplying as fast as mechanically possible. It's exactly like stealing a car. The Hedgehog pathway is the ignition key, or, the hot wire. You use Hedgehog to start the engine and initiate the complex process of getting the car moving.
Right.
But once the car is speeding down the highway at 100 miles per hour, which represents the late-stage TNEPC, it doesn't need the ignition key anymore. The key is irrelevant. The cancer just slams a brick on the gas pedal, and that brick is the Myc pathway.
And this Hedgehog to Myc switch explains so many past clinical failures and oncology trials.
This raises an important question. If doctors try to use a Hedgehog inhibitor drug on a patient who's already in late-stage NEPC, it would be completely useless, wouldn't it? because the key is already out of the ignition.
It would be entirely ineffective. The cellular dependency has completely shifted. This dynamic switch means that in precision oncology, timing is just as critical as the drug itself.
So you have to hit it early.
We have to hit the Hedgehog pathway early, during the specific window of transition before Myc takes over and drives the rapid, unstoppable proliferation.
So to wrap up this mechanism, the cancer utilizes a dynamic, stage-specific signaling switch. It uses the Hedgehog pathway as the hot wire to drive the initial transdifferentiation, but then swaps to the Myc pathway to act as a brick on the gas pedal.
Exactly.
Does knowing that a cancer literally swaps out its internal operating system midway through its lifespan completely change how you view targeted medicine?
It proves definitively that a static, one-size-fits-all approach to a dynamic disease will always fail.
If we try to synthesize everything we've explored today from this NPJ study, what's the ultimate takeaway?
The overarching truth is that a tumor is not a static lump of rogue, isolated cells. It is a highly dynamic, constantly adapting, whispering community.
Wow.
It senses our pharmaceutical treatments, it communicates with its neighbors through paracrine signaling, and it fundamentally alters its lineage to survive. The key to beating it isn't just diagnosing what it is today. It is about intercepting what it is actively trying to become tomorrow. And with the 6-gene alarm system and our new mechanical understanding of the Hedgehog to Myc switch, we finally have a high-resolution map to do exactly that.
It is a profound shift in perspective. We spend so much time in medicine trying to destroy the destination, the final, deadly, aggressive tumor. But what if the future of curing cancer lies entirely in treating the transition? What if we just stop the costume change in the alley before it even finishes?
Intercepting the transition is the next great frontier of precision oncology.
Thank you for joining us on this deep dive into lineage plasticity and the incredibly complex survival tactics of cancer. As you go about your day, I want you to think about how this kind of cellular peer pressure might be happening in other inflammatory or autoimmune diseases you've heard about.
It's definitely worth looking into.
We highly encourage you to explore the biological concepts of transdifferentiation and lineage plasticity on your own. It's a fascinating rabbit hole. But before we go, I want to leave you with one final thought to mull over. If a tumor can learn to rewire itself with brain neuron genes just to survive our best drugs, what other hidden, terrifying intelligence is locked inside our biology, just waiting to be pressured into the light?